Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A-Level Biology Unit 4 potential questions for any Unit 4 biology paper. Let's begin with Topic 5. Topic 5, Energy, Ecosystems and Climate Change. Here you need to learn about the ATP molecule. You need to learn to draw the ATP molecule, including 1 adenine, 1 ribose, and 3 phosphates. You need to learn to distinguish between autotrophs and heterotrophs. Remember, heterotrophs feed on plants as well as other organisms, while the autotrophs use CO2 in order to generate their own food. Remember, ATP is made from ADP and PI through a condensation reaction. This is an enzyme-catalyzed reaction, and therefore changing the conditions for a reaction can alter how this reaction proceeds. You need to know that ATP is broken down to ADP and PI by a hydrolysis reaction. This hydrolysis reaction involves adding water, so both these reversible reactions are catalyzed by ATPase, which is the enzyme for that reaction. Continuing on, you are required to know about the chloroplast as well as chlorophyll. Remember, a chloroplast is an organelle where photosynthesis takes place, so you need to learn to draw and label the components of a chloroplast. This question is very common, so learn to position the thylakoids, the inner membrane, the outer membrane, the stroma, and sometimes you are required to put in some circular DNA. Usually this is a 3-mark question. You need to remember the functions of each component in the chloroplast. An example, why is the inner membrane folded? Why are the thylakoids the way they are positioned? So you need to know that. You also need to remember that chlorophyll is positioned inside there because that is where these photosystems are found. So they can ask questions about adaptation of the chloroplast for its function. You need to remember about the various chlorophyll molecules. Remember chlorophyll A, chlorophyll B, carotenoids, and how the ability of these molecules to absorb sunlight in different wavelengths could be beneficial for a plant that possesses the different types of chlorophyll. So they can give you data about absorption spectra as well as action spectra, and they could ask related questions. Ensure that you fully understand the information in a question before you answer, because if the question is confusing, you may get it wrong. I recommend that you put great emphasis on using chromatography to separate chlorophyll molecules. Questions could be brought about this using chromatography as well as RF calculations. You need to focus on this. Moving to photosynthesis, you need to learn the details of photosynthesis, the light-dependent reactions as well as the light-independent reactions, the process of light-dependent reactions leading to reduced NADP as well as ATP production. Here, please focus on cyclic photophosphorylation and non-cyclic photophosphorylation. And remember how to compare and contrast these processes. Remember, one leads to the formation of reduced NADP, while both processes lead to the formation of ATP. I recommend you learn the details of each. In cyclic photophosphorylation, the electrons are recycled, they return to the photosystems, while in non-cyclic photophosphorylation, the electrons are taken away by the NADP that has been reduced. You need to know about the fate of NADPH and ATP in the light-independent reactions. So here you must remember how Rubisco is used as an enzyme in the fixation of carbon dioxide. You need to know about the conversion of Rp into Gp. You need to know about the conversion of Gp into GALP. They could ask you how GALP is used to make proteins. Remember, you add nitrates and then proteins or amino acids could be produced, and those amino acids could be combined in condensation reactions leading to the formation of proteins. They could ask you how the GALP is converted into starch and stored. You need to know about the transportation of GALP. An example, it could be converted into sucrose, and then the sucrose could be moved down through the phloem into the roots where it could be stored. So details here are required. They can ask questions connecting the changing conditions like temperature, sunlight, carbon dioxide concentration, and how these changes affect the rate of photosynthesis. So fully understand detailed explanation of how changing these conditions affect the enzyme-controlled reactions, including those of the light-independent reactions. Moving on to ecology, you need to learn the following definitions. You need to learn what an ecosystem is, a habitat, a population, a community, as well as a niche. You need to learn to explain how abiotic factors like temperature, sunlight, wind, rainfall, water availability, oxygen availability, as well as edaphic factors, and the biotic factors like disease, predation, finding a mate, competition for territory, and so on. You need to know how these factors affect the abundance and distribution of organisms within a specific ecosystem. You could also be given data in the form of graphs or tables, so ensure you fully understand the question before you attempt to answer it. Questions could come about succession, and here they could bring images, maybe a graph or maybe images like in tables to show you how a specific area has been transformed due to succession. Remember, succession can be primary succession or secondary succession, and you need to include the details of how the pioneer species come and how those pioneer species change the conditions in order to allow other organisms to come. Change conditions could be change in pH, water availability, salinity. Sometimes the environmental changes like temperature as well as sunlight could affect how succession occurs on a specific piece of land. You need to remember the climax community and how it's going to be self-sustaining and how any changes on the climax community could cause a change in the organisms that are thriving in a specific area. You need to learn about the three main patterns of distribution of organisms. Sometimes organisms can be uniformly distributed. You need to know why they could be that way. If they are clumped, maybe there is insufficient resources or they are running away from a common enemy or predator. And if they are randomly distributed, it could be because the resources are very abundant. You need to learn the detailed description of using quadrats. Remember, quadrats come in different forms. They could be frame quadrats, point quadrats, permanent quadrants, as well as area quadrants. So you need to know how these quadrats are used and when they are required. For example, when do you use a frame quadrant? When do you use a point quadrant? When isn't it required? You need to know the details of that. You need to know also the details of using transects in finding out the number of organisms in a specific area. Continuing on, methods of measuring abiotic factors. This could be common in Unit 6 as well as questions that require you to design experiments. Like if you're measuring the dependent variable, you will tell the examiner, I measure the pH using a pH meter, I measure the light intensity using a light meter, oxygen levels using a digital oxygen probe, as well as temperature using a thermometer. So please keep in mind these things when you answer your Unit 6 questions as well. About the statistical tests, these are rare in Unit 4. However, I recommend you do not leave any stone unturned. Please revisit every part. I know this is common in Unit 6, but do not take any chances. Learn to state the null hypothesis and to calculate using these equations, the statistical tests. You need to learn to comment on the validity of the data using error bars or any calculations that you have carried out. I recommend you do that. Moving on to environment and climate change, most of the questions asked from this chapter are going to be about data, so remember to practice with graphs as well as data tables from past papers. On that note, you need to remember the definitions: what are producers, what are primary consumers, secondary consumers, as well as tertiary consumers. You need to know the roles of each component, including decomposers within either a food chain or a specific ecosystem. They could ask you about pyramids of numbers, pyramids of biomass, as well as pyramids of energy. Please ensure you fully understand this. On the note of energy, they could ask about energy losses in a specific food chain or in an ecosystem. This is a commonly asked question in connection to data questions. Remember, energy can be lost due to chemical reactions or metabolic reactions that go on within organisms, or sometimes some food substances may not be digested or they may be excreted out, so those can lead to energy losses within a specific food chain. So remember also net primary productivity, that this is equal to gross primary productivity minus the energy used up in respiration. A lot of questions come from tables and data, and some explanations may require you to refer to this kind of information. They could ask about the carbon cycle. Please remember the role of decomposers in the carbon cycle because these are used in the recycling of carbon dioxide, which is later taken up by the plants in the process of photosynthesis. And when we talk about decomposers, these mainly include bacteria as well as fungi. You need to learn the details of the greenhouse effect as well as greenhouse gases. Please remember which gases they are, like carbon dioxide, methane, water vapor, and so on. You need to learn the definitions like anthropogenic climate change, what is climate, what is weather. Also, you need to fully understand the methods for looking for evidence. Many data questions are going to be connected to this, so they can give you temperature records. They could use isotopes of oxygen, maybe in ice cores, in order to determine the temperature records. There is evidence from dendrochronology using the tree rings as sources of evidence, as well as peat bog data. Now, using peat bog data is very common, so please understand they could give you information about pollen data and ask you even a question relating to succession so that you can determine which species of plants have occupied a specific area over a longer period of time. This has been asked previously. Please fully understand the difference between a correlation and a causation in data interpretation. If you look at data more so relating to climate change, the evidence given, does it show a correlation or it shows a causation? You need to fully understand that. You need to know that climate change is linked to global warming as well as the rise in global temperatures. You could be asked about the consequences of this rise in temperatures on processes that are biological, like photosynthesis and so on. Other consequences of global warming include seasonal changes, melting of the ice caps, increased sea level, flooding, destruction of habitats, extinction of species, displacement of organisms, and so on. Please make sure you can reason in relation to any of these in case data is given to show maybe displacement, extinction of species, movement of organisms from one area to the other, or if it's increased in relation to seasonal changes. Ensure you fully understand. Now, on that note, I'm going to talk about climate change and evolution. Remember, climate change can cause allopatric speciation, which is due to geographical isolation, and it could lead to sympatric speciation, which is related to organisms being in a specific area or similar area but having reproductive isolation. Linked questions could ask you how to overcome the problems brought about by climate change. So remember to talk about using sustainable resources like biofuels, as well as carrying out reforestation to plant more trees in order to preserve specific habitats. Topic 6, Microbiology, Immunity and Forensics. Here we'll begin by looking at bacteria and viruses. Please remember that all bacteria have a cell wall made of peptidoglycan. They have 70 S ribosomes and a nucleoid, as well as a cell surface membrane. Practice to draw and label basic components of a bacteria as above and include others like the pili, the flagella, the slime capsule, mesosome, and I recommend that you remember the functions of all these components. Please remember the major components of a virus, like they have nucleic acids. Some viruses are RNA viruses while others are DNA viruses. You should remember that some have a protein coat. They have surface proteins, which could be antigens, and these are used for infection. And they also have a nuclear envelope. Some of them have nuclear envelopes and others do not. So please remember the different types of viruses. Like I said already, some are DNA viruses, others are RNA viruses, and we have a special classification we call the retroviruses. Remember examples of each classification, like HIV is a retrovirus, lambda-phage, DNA virus, and then we have Ebola and influenza, which are RNA viruses. Please remember that some viruses have specific capsids that have specific shapes, like TMV and Ebola have the helical, some have the complex like the lambda-phage, as well as the polyhedral like we have in HIV. They could ask you to compare and contrast the structures of viruses and bacteria. Ensure you fully understand those. And please remember the details of the lytic and the lysogenic pathways in the viral life cycle. Remember, the lytic pathway has a way of destroying cells that have been infected in massive numbers, and if these are immune cells, the immune system is going to be broken down or brought down or weakened greatly. This is evident in HIV infection because remember the HIV virus infects T cells, and if these cells are broken apart or destroyed in large numbers due to the lytic pathways, the immune system is going to be weakened a lot. I recommend that you put great emphasis on the description of the details of the life cycle of a retrovirus, like HIV. Remember that it contains reverse transcriptase, and this converts RNA into DNA. Again, remember HIV is a special type of RNA virus. It has RNA, but the reverse transcriptase is going to use it to convert the RNA into DNA, and that DNA is going to be integrated into the genome using integrase enzyme. When we go to growth of bacterial colonies, please know the shape of the bacterial growth curve as seen on page 94. Remember, it has a sigmoid shape and you need to know the reasons for each part of the shape or each phase of the curve. We know we have the lag phase. This shows the bacteria or microorganisms adapting to the new environment. We have the log phase. This phase is where we have, or you call it sometimes exponential phase. That is where there is the rate of reproduction is almost at a maximum. There is the stationary phase. In this phase, the death rate is equal to the reproduction rate. And then we have the death phase, where we see the death rate is greater than the reproduction rate. And this could be due to accumulation of waste, decrease of nutrients, as well as competitions for space. Practice calculations relating to the growth rate constant. When we go to microbial techniques, please put great emphasis on knowing how to culture bacteria and the right conditions that are required. We need to have the optimum temperature, the right pH, nutrients, as well as the oxygen. Great care has to be taken when handling these microorganisms, when culturing them, as well as disposing them. We need to use aseptic techniques to culture, and using a sterile loop is one of them, or a sterile environment, as well as wearing gloves. We could obtain sterile surfaces by using either sterile materials to lay down on the surface before we carry out the experiment, or we could use specific alcohols in order to sterilize. Culturing can be done on solid medium, like agar, or it can be done on nutrient, which we call the broth, the liquid medium. So please practice descriptions of these two methods of microbial culture. And a selective medium can be used to ensure that the pure culture has been obtained. The selective medium could have a specific nutrient. It could have some medication like antifungal or some antibiotics, as well as other components that could be chosen based on the desires of the person culturing. The next part is about measuring the growth of bacteria. Here, I prefer you master description of cell counts using a hemocytometer. And please remember the total number of cells per centimeters cubed is equal to the cells counted times 10 to the power of 4. I recommend that you practice description of optical methods as detailed on page 100 and go through dilution plating as a method of counting bacteria. The details are in your textbook. Sometimes for fungal and other microorganisms that can be larger in size, we could use dry mass, obtaining the microorganisms, drying them completely to remove all the water, and then weighing the mass, or we could try to calculate the area. So related questions about the effect of antimicrobials, as well as the effect of changing growth conditions onto the growth rate of the microorganisms can be asked. Please do not forget about using the zone of inhibition, the area, as well as the mass of the dried microorganisms. I recommend that you go through the spread of microorganisms, like through ingestion, vectors, fomites, inhalation, inoculation, as well as direct contact. Please revisit barriers to infection, like surfaces or epithelial defenses, including the skin. Remember, the skin has tough keratin that is hard to penetrate, so many microorganisms are blocked out from entering into our bodies. And also our skin produces sebum, which has antimicrobial properties. We also have natural skin flora that compete with the pathogens for space and nutrients in order to prevent us from getting infection. Please remember that we have surfaces that produce mucus, and this contains lysozymes that can digest the microorganisms. We also have HCl in our stomachs that can destroy some of the pathogens that we could ingest. And finally, we have gut flora that can outcompete the pathogens for space and nutrients in order to keep us safe. You need to focus on how bacterial toxicity causes disease. Here, focus on endotoxins. Remember, these are lipopolysaccharides, and the lipid part of these lipopolysaccharides is the toxic one. Remember, they can be found on gram-negative bacteria. With exotoxins, these can come from both gram-positive and gram-negative bacteria. Bacteria also cause disease through host tissue infection, like in the case of tuberculosis. When we go to immunity, you need to remember we have leukocytes, which include granulocytes as well as agranulocytes. Granulocytes have granules, and these include the lymphocytes as well as the monocytes. And the agranulocytes include the basophils, the neutrophils, and the eosinophils. On this note, they can ask you about non-specific immune response. Remember, it includes inflammation, fevers, phagocytosis, as well as interference. Inflammation is caused by basophils as well as mast cells producing histamines, and these histamines are going to trigger inflammation. Fevers are caused in order to increase the temperature so that the growth of the pathogens is slowed down in order to increase the rate of phagocytosis. Phagocytosis, of course, is going to be caused by neutrophils as they release lysozymes onto these pathogens. However, we need to know that neutrophils cannot carry out phagocytosis for longer periods of time because they cannot renew their lysozymes. Interferons are produced due to invasion by viruses. So they are produced by cells that are being invaded by viruses in order to fight off the infection. When we go to specific immune response, remember this has lymphocytes as well as monocytes. Our immune system has many aspects that are helpful. It can distinguish self from non-self. It has immunological memory. It is specific and it can recognize many different antigens. You need to remember that lymphocytes are divided into B cells and T cells. And remember the details of the humoral response, including the details of T cell activation, production of T memory cells, B cell activation, B memory cells, B effector cell differentiation into plasma cells, and how these B plasma cells produce antibodies. In connection to antibodies, they could ask about protein synthesis. So revisit, remember antibodies are proteins and the process through which they are produced is protein synthesis. So they could ask a question in relation to that. Remember that antibodies cause agglutination, neutralization, as well as opsonization. This question is very common in past papers. Remember to detail the cell-mediated response, meaning production of activated T killer cells, and how they produce bufferins to cause lysis of the membranes of infected cells in order to destroy them. They can ask about primary immune response as well as secondary immune response, so they could ask to compare and contrast this. When it comes to artificial immunity, remember we have artificial active like vaccination, we have artificial passive like transfusion, we have natural immunity which includes natural active like when you're infected with a real pathogen, as well as natural passive like breastfeeding. Lastly, on this note, remember that there is no antibody production or memory cell formation with passive immunity. When we get to antibiotics, remember they can be bacteriostatic or bactericidal. They could bring a question on how antibiotics work. Remember that bacteriostatic antibiotics inhibit growth and production of bacteria by interrupting processes like protein synthesis as well as metabolism. Bactericidal can kill pathogens by causing damage to their cell membranes, preventing the cross-linkage formation in cell wall, and so on. Continuing on to antibiotic resistance, this is a common question when they talk about evolution and race between the scientists and antibiotic resistance among pathogens. So please remember to connect evolution to pathogens. They could talk about multi-drug resistant pathogens. Again here, the role of variation and natural selection in drug resistance could be connected. This is a very common question as well. They could bring about hospital-acquired infections and practices that can be carried out to avoid this, like isolating patients, washing hands, checking patients for specific drug-resistant pathogens before admission. They could ask about overcoming drug resistance, like restriction of prescription that is carried out by the doctors, ensuring the medication is taken appropriately, that is in relation to the patient, and then carrying out good hospital practice, that is in relation to the caregivers within the hospitals. The next part is microorganisms and decomposition. Ensure you learn decomposition is carried out mainly by bacteria and fungi. However, the conditions have to be perfect for decomposition to occur. Conditions like temperature, moisture content, oxygen, pH are vital in either fastening or slowing down the rate of decomposition. They could ask about details of how this decomposition takes place. Ensure you fully understand that these pathogens release enzymes. Remember that these pathogens release enzymes onto the substance they desire to decompose. Then digestion will occur externally and the substances that are produced are going to be absorbed as nutrients to these microorganisms. Remember to revisit the factors that affect how quickly the body decomposes. Remember mode of death, the fat levels, temperature. The higher the temperature, the higher the rate of decomposition until the optimum is reached. They could talk about surface area to volume ratio of the body, then whether the body was covered or not. Questions can be asked, and these could be connected to data or graphs, so ensure you fully practice with past papers. The next part is estimating the time of death. They can talk about the methods for investigating the time of death, like body temperature, When somebody dies, the body temperature is slowly going to decrease until it reaches the ambient temperature. So they could use that in order to determine when the person died. They could use rigor mortis, but this has advantages and disadvantages, or when we can use it and when we cannot. If a body has been dead within the first few hours, we may not be able to use rigor mortis, and if the body has been dead for quite a while, maybe 2 days, we may not be able to use rigor mortis. So also we could use forensic entomology. This is more effective when some time has passed on. You need to remember to describe these methods in detail because actually data can be brought. They could be graphs showing how the body temperature decreases. They could be graphs showing how the change in rigor mortis or stiffness of muscles. Information showing the specific species of flies that have been found on the body. That could be connected to other questions. Remember to explain situations in which it is suitable to use any of the above methods to determine the time of death. Required for this will be given, so please revisit past papers on this topic. Understand why scientists take some sample of eggs from the decaying or decomposing body, put them into the lab under specific conditions in order to hatch the species of flies so that they can study which species they are and study their life cycle in order to estimate the time of death. Remember to practice calculating hours in case the life cycle is given and you are required to work backwards in order to estimate the time of death. A question similar to this has come previously. Questions could be asked about DNA profiling. Ensure that you fully understand the details for PCR, how it is carried out, what is required. Like you need DNA, you need the Taq polymerase, you need primers, you need nucleotides and the suitable buffer. You need to learn this as well as the description of the process. You need to remember how DNA samples are prepared for gel electrophoresis. You need to go through the details of gel electrophoresis, including preparation of the sample, preparation of the gel, feeding the sample into the gel, and which suitable dyes are used and why they are used. Please remember that restriction enzymes are used in order to fragment sections of DNA, and the same restriction enzyme is going to be used in case specific sections are required to be cut at the same position. Remember how gel electrophoresis data can be interpreted. Remember that thicker bars show a higher concentration, and the bars that appear closer to the negative electrode are going to show the heaviest DNA. Bars closer to the positive electrode are going to show the lightest DNA because that DNA would have moved the fastest or the farthest from the origin. On this note, questions could be brought comparing specific DNA segments from one person and another person or one organism and another to see similarities as well as differences. Please ensure you fully understand how this DNA profiling can be used to tell similarities as well as differences among organisms. To do that, you need to collect the DNA sample, you need to cut out sections of DNA you're interested in, and then you need to load the samples into a gel and then observe the bars that are produced in order to interpret results to see if there is close relationship or there is no relationship at all. This brings us to the end of these potential questions. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.